ones should be okay. I mean, there's no reason, but just I'm afraid that my connection is not very good, so I don't know whether it will be a bit slow. You see okay?
Yeah, we see it running. That's good.
Okay, perfect. Thanks. I'm done.
Right.
So who's first actually?
Jorge, you'll go first if that's all right.
Okay,
as
perfect.
the junior. Mina, do you want to have a go?
Yeah,
the screen?
I'll try one of.
Yeah. Thanks for the bio sketch, by the way. Sorry, I forgot to ask for it.
Yeah, no worries. I'm glad that you got it.
Thank you.
Let's see. Maybe. Okay, everyone seeing full screen?
Mm-hmm.
Yeah.
Great. Okay. If that
Yeah, we see them running.
Great.
That's good. Thanks a lot.
Great. Thank you.
Oh, bang on time. Perfect. You're YouTubing, right, Jen?
Yes, we are streaming live on YouTube.
Let me just tweet it and then we can get going.
Okay. Hi to everybody who's here. Welcome. We're just doing a few last minute setup things, sending out notification tweets. We'll get started in 2 minutes. Minna, how's everything in Utah?
It's pretty rough here.
Yeah, I mean, I've been seeing you guys on the news a fair amount, but I don't know anyone closely there. So
yeah, unfortunately, we're making national news.
Yeah.
Yeah, it's not great here, but not a lot being done about it. So it's been a rough go.
Are you guys? You're still working, or like still going into? People are still going into the lab and all.
The university is operating at about 50%. Yeah, to maintain distancing and everything. So we're still there, and we'll probably be there for a while. I don't anticipate moving into the next phase for at least a few months as we get through flu season.
The next phase being more open.
Yeah, more open. Yeah.
Okay, but you also don't anticipate like more shutdowns there because there's a lot of.
It's possible. The university actually is not allowing students to come back after Thanksgiving, but that's our undergrad campus. So everyone, it'll be fully remote after Thanksgiving, American Thanksgiving. Yeah.
Yeah, I'm hearing a lot of. Right. Well, who knows? We're in the unintended consequences phase of things, right?
Exactly. Yeah.
Well, I'm sorry that your state is making national news for
not
the very best of reasons.
Yeah, usually we make, people talk about our national parks and our ski season.
Right, which you still have a beautiful state.
We still have, yes. But actually all of that will be changing. The ski resorts are open, but not operating at full capacity, which is good. Yeah, to keep people safe.
Yeah, I imagine the skiing itself is pretty safe, but the lodges might not be.
And the lifts, yeah.
Oh.
Yeah.
Shall we get started? Okay, so hi everyone, thanks for tuning in. So we've got 2 speakers today and we'll just crack on. Same as normal, so any questions, pop them in the chat or write a question if you'd like to ask it yourself. So our first speaker is Jorge Barbazan. Jorge did his PhD in the University of Santiago de Compostela in Spain in the field of circulating tumor cells and metastatic liver colonization. He then moved to the lab of Daniela Vinovics at Institut Curie, Paris, for his postdoc, where he focused on cancer-associated fibroblasts and their mechanical interplay with cancer cells during tumor development. He's recently become an independent researcher in the Health Research Institute of Santiago de Compostela, and he is developing several lines of research on translational cancer mechanobiology, and he's going to be presenting some of his postdoc work from Daniela's lab today.
Cool. Thanks, Adam. So I will share my screen. Okay, let's see. Hope it doesn't crash. Okay, good. Can you see it? Can you see my mouse as well? Perfect. Well, so thanks very much. Well, good afternoon to everyone. And thanks very much to both Jen and Adam for the invitation to speak here today. I think that's a pleasure for me to talk in such a great meeting that you kind of this seminar series that you managed to organize over the last months. I think it became a really good forum for cell biology and also cell migration. So today I would like to share with you a project that I did when I was a postdoc in the lab of Daniela, which I hope it will be soon in full detail in bioRxiv, and which is a project that will be in very close collaboration with the lab of Xavier Trepat, and with a student, what at that time Carlos was a PhD student in his lab, and now he's a postdoc in Daniela's lab. And that was a really wonderful collaboration. So half of the work, I mean, that I'm going to show you here today is Carlos. So, and the project is about how the mechanical interplay between the stroma and the cancer cells actually regulates the architecture of tumors and how tumor cells behave in the tumor. So I don't think I have to go into details on this, but just to give you a brief introduction, normally tumors, and here I'm talking about solid tumors that develop from an epithelium. Once this particular sort of cells that will acquire some mutations, so will start over-proliferating, and they will eventually be able to break the basement membrane and invade into the surrounding stroma. And some cells will be able to make it to the circulation, and those cells will be able eventually to make metastasis on the distant side. So to go through all this sequence of events, cancer cells, they normally have to acquire some specific properties that have been summarized within the last years in what we call the hallmarks of cancer, which include some stuff like, for example, like sustained proliferation, avoiding apoptosis, or just like activating metastatic or invasive pathways, or like increasing angiogenesis. But one of the things about this properties that have been defined as very like cancer cell centered, and they kind of avoid to include in them like how the interplay in between cancer cells and other cell types in the microenvironment that are closely surrounded by them. And so the last year's much more attention has been paid to actually how the physical stresses that the cancer cells feel from their outside, which are mainly like created by other cells in the microenvironment, could actually regulate all this process that we can see here. And I took this picture from a very recent review, and it's a very nice review in Science, which actually they describe that apart from the hallmarks of cancer, we have the physical kind of hallmarks that will define how tumors behave. And actually normally kind of includes the solid stress, this fluid pressure, the microarchitecture of the tumor, and how the stiffness also regulates how tumor cells behave. So just focusing on what happens with solid stress, which is normally attributed towards the growth of the tumors within, like, of the tumor cells because of their uncontrolled growth pattern, that will eventually push against the barrier which is around tumor cells, which is normally composed of the extracellular matrix and the stroma. And this has a property of a viscoelastic material. So we'll try to resist this
Okay, I think Jorge's gone for everybody. Yes? All right, let's give him maybe just a minute to, oh, he's back. Hey, Jorge, are you still there? Can you hear me? You're muted. I don't know if he can hear me here.
Okay, was that all of us?
Yeah, I think that's everyone. Jorge, I think you're muted still.
It's okay?
I know we can hear
me?
Yeah, we can hear you.
But your slides are gone. Yes?
So it happened only to me, or it happened to everyone?
To everyone.
Your slides disappeared for everybody. And you were
okay.
So maybe back up about
because
a minute if you're gonna restart.
Okay, I actually stopped seeing you all.
Oh no.
Yeah, so I don't know if it's my internet connection or. Okay, so should I continue over here?
Can you see us and hear us?
I hope it's not gonna happen again. Yes, I can see you and
Okay.
hear you perfectly.
Okay, yeah, I think you can carry on now. Maybe
Okay.
just back up 30 seconds or so. Thanks.
Okay, so I was telling you that there's this physical properties of that surround the tumor that will actually determine how the tumors behave, and I was talking about this solid stress that will has very high like important consequences on tumor development that will actually, for example, be compressing blood vessels within the tumor, just creating this necrotic core and this hypoxic regions in the tumor, and will actually also influence a lot how the therapeutics, for example, can be delivered into the tumor just by because of this fluid pressure that can be generated inside the tumor. So, however, normally this solid stress is attributed only to the active growth of the tumor, and the barrier, like this stroma, is considered rather a passive structure. So this stroma is normally considered of many cell types that are included in the tumor microenvironment, and amongst them, the majority of them are composed by what we call cancer-associated fibroblasts, that are just myofibroblasts that become activated from normal resident fibroblasts, or just from other like precursors that come from many other cell types. And at the end of the day, they create this desmoplastic reaction, which in some tumors, they can even span the majority of the tumor, like in pancreatic tumors. So these CAFs, they will have a physical activity over tumor progression, and some labs, they have demonstrated that they can actually physically remodel the extracellular fibers. I mean, this ECM around the tumor, creating this parallel highways that cancer cells, they can use to escape and invade into the surroundings, or they can even be pulling physically cancer cells out of the tumor. But other labs, they have showed the opposite, and they actually, and this was in pancreatic cancer, that actually showed that CAFs, they can create this very dense and thick capsule of stromal capsule that will actually prevent the expansion of the tumor cells. However, even if it looks that this formation of this capsule is rather important, actually this capsule is the one that is restraining and this growth of the cancer cells, and this generating this solid stress that I was telling you about a minute ago. It's not really known how this capsule is formed, and also it is not known if it actually has an active mechanical role over cancer cells. So to start understanding these two questions, we start having a look on tumors that we have on a mouse model that we have in the lab, whether in which we can generate intestinal tumors by overexpressing Notch and knocking down p53 on this, specifically on the intestinal epithelium. And 1 year later, more or less, these tumors will become invasive, and they will look like this, with cancer cells in green and the surrounding stroma in expressing tomato, which I labeled here in magenta. So tumors, they look more or less like this. This is a rather big chunk of the tumor, where you can see this big capsule of stroma that surrounds the whole core of the tumor, and it's kind of infiltrating. And if you have a closer look, you will see how the microarchitecture of the tumor, it reminds again to this capsule that we showed before, with fibroblasts surrounding this cluster of cancer cells. And this is something that is kind of well conserved. We see the same in mouse colorectal tumors, in patient tumors. And if you take primary cells spontaneously, that spontaneously go out from a piece of tissue that comes from a patient, they will spontaneously organize like this, with cancer cells again in the center and CAFs that goes surround. So we wanted to see what actually happens when we put this co-culture of these two cell types, and we see what happens with over time. So in this case, we have a cluster of cancer cells over here, another one over here, and a small one here, and the rest is CAFs. And the first thing that you observe is that CAFs, they have this property of aligning towards the boundaries of the cluster of cancer cells on an intrinsic property that is called nematic ordering. And if you see what happens over time, it looks that the CAFs are kind of creating this boundary that instead of promoting the cell, taking cells out of the tumor, is kind of restraining and pushing them back. And it looks like they're doing that on a pretty active way. And whereas more obvious is if you have a look on the left part, where this, in particular geometries of the cluster of cancer cells, CAFs, they can generate a ring that closes on top of the cancer cells and squishes them. So here you can see another representation of this. In this case, we label the cells for more visibility. And in this case, you can see how this ring of CAFs is actually closing on top of cancer cells and squishing them. And if you see it better, if you follow this 3D rendering where the CAFs are labeled in red and cancer cells in green, and see how they're jumping on top of them and they're generating this compressive structure over there. So far, I'm always talking about compression, but we don't really know if there's any compression, active compression going on there. So for this, we teamed up with the lab of Xavi, and we started to do very simple experiments in 2D measuring traction forces. So what happens when you follow what happens with the forces in this co-culture system, where you see the spring forming and squeezing cancer cells, what happens is that we see the formation, the accumulation of this traction forces that are radial and pointing towards the inside, which are suggestive of compression. So we can kind of classify these forces in two different types: tangential, which don't really show up. However, radial forces, they accumulate over time, and they switch from a outward pointing forces in the beginning of the movie that corresponds to cell crawling, trying to penetrate the cluster of cancer cells. And as soon as this compressive ring is able to form, they will switch towards inward pointing traction. So basically they will form a force tree, as you can see here, that will actually generate the force in the area where the cancer cells are squeezed. And after, sorry, can I point here? Yeah, so after this force will be transmitted to the CAF smaller layer, to the substrate. And what we see here is that right at the edge of the cluster of cancer cells, we see this accumulation of radial forces where nothing is really going on with the tangential ones. So, so the generation of this force for compression comes from obviously actomyosin. So you see that this compressive rings, they express a lot of F-acting and active myosin, phosphomyosin, and it happens the same in vivo. And actually, what happens is that if you inhibit contractility and inhibit myosin using blebbistatin or Y27, what happens is that this compression doesn't happen anymore, and the ring completely relaxes, and this is the moment that cancer cells, they use to expand. And this is what we see here in the quantifications, where you actually see how in both conditions cancer cells start expanding upon addition of the drug, and the forces, they go like virtually to zero. So, and this is what happens in vitro, and we wanted to see what is actually going on a more relevant system. So we switched to an in vivo model using the same mouse model as before, and we combined it to another mouse model where we can specifically knock out myosin 2A on cells that express alpha-SMAP. So this is a very well characterized marker for CAFs, but it's not 100% specific because it's also expressed in cells like pericytes or other cells that express this molecule. So we wait for tumor formation in this type of mice, that we call it the donor. And once the tumor forms, we generate organoids and we take the bone marrow from these guys and we transplant it in irradiated recipients that we kill basically the immune system and we change it with the immune system of the other guys. So this will make them immunologically compatible. At the end of the day, once the immune system is reconstituted, we implant subcutaneously the tumor organoids from the donor mice, and we wait for tumor development, and in some mice, we just start injecting tamoxifen. So at the end of the day, what we will generate are tumors that are generated on a compressive or a non-compressive environment. And this is how the tumors look like in terms of colors, because also the CAFs that will be knockout for myosin, they will switch from tomato to membrane GFP, and they will start being green. So this is very useful for us to identify the cells that are being knocked out. So this is a first impression of how the tumors look like. This is a control, and in green you have the cancer cells, and in red you have the stroma. So what you can first see, and this is a kind of a big part of the tumor of several millimeters, where you can actually see is that there is this very established compartmentalization of the stromal part versus the tumor part, with this capsule that is being formed over here, and several types of the tumor, and several parts of the tumor. And what actually happened in the myosin knockout is that this organization gets completely lost, and there's a complete mixing of the two cell types. And this is even seen better if we go and see what happens with the fibrillar structures just by imaging on second harmonic. where we basically are showing what happens with collagen structures. And you see basically the same. This very thick and dense bundles of collagen that form the capsule again, separating stroma from tumor part that actually doesn't exist anymore or gets very much disorganized in the case of the myosin knockout. And this is just assumed just to show you what I'm just gonna conclude from this part, which is basically that contractility is necessary for the formation of the capsule that you can see here on the left. And upon abrogation of contractility in calves, there's a complete disorganization of the architecture of the tumor. So just to try to understand what happens with the forces in between these two cell types, we started to do laser ablations on the fresh tissue that we just cut on the vibratome, and we put it on the 2-photon. And in this case, we normally do it on typical structures like this, where you see this cluster of cancer cells surrounded by the stroma, and we'll just be ablating the boundary in between the two cell types. So what we actually see is that as soon as we ablate, there's a very fast expansion of the tumor cells. And you can even see better if we quantify this using PID. So we basically quantify what happens with the cells close to the wound, close to the cat, or far away as possible from the cat. And what we see when we quantify this is that the displacement of the cells is much higher when you're close to the area where we are actually releasing the stress. And this is actually directional, so the cells are going towards the cat, where the tension has been released. So this indicates that the cancer cells are actually under compression. What happens with the calves? So for this, we switch to a slightly different type of cut. In this case, we cut perpendicular to the edge of the cluster of cancer cells, and we basically see that upon cutting, and you can follow here in the yellow arrows, calves, they recoil on the opposite direction against the cat, whether cancer cells, they still go towards the cat, while when the compression, the tension of the calves is released. And here is just again the same quantification as before. And what you can see here is that cancer cells, they will go towards the cat once we perform the cut, and calves, they will go against from the cat. So this is again dependent on myosin 2 activity, because if we just treat the tissue for a couple of hours with blebistatin, we just see that there's a high inhibition of or reduction of displacement, and the cells that are actually moved, they don't do it on a directional way anymore, and they actually it happens the same when we analyze what happens in the case of the myosin knockout. So that indicates that cancer cells are under compression by calves, and that depends on myosin. So this is just to verify this in vitro on a much simple system, where we, in the same system that I just told you before, where what we see is that everything actually happens on a much more exaggerated way, where we see that's this immediate expansion of the cancer cells that goes together with the recoil of calf, and the direction of movement of the cells is the same one that I just showed you before. So we next wanted to understand if this has any relevance in kind of the biological properties of the tumor cells. And for this, we decided to address the nuclear localization, or the localization of YAP, which is a very well-established mechanosensor, and it will switch in and out the nucleus depending on the mechanical stress that the cell is feeling. So for this, we do this correlation with DAPI signal, which will be +1 if the DAPI and YAP correlate, so it means that YAP is in the nuclei, and will be -1 if YAP is completely cytoplasmatic, and will be 0 or intermediate levels depending on the change between cytoplasm and nucleus of the cells. And what we actually see in vitro is that in the control situation, YAP is basically cytoplasmic, and as soon as you use CAF, but they are not contractile because they lack myosin 2, YAP becomes more nuclear. And the same happens if you actually use blebistatin. So that actually indicates that this mechanical compression that the stroma is exerting onto the tumor, and actually, well, maybe I start first here, because we actually see exactly the same pattern in vitro. And this actually means that this mechanical compression that the calves are exerting onto the tumor is just not a simple, a compression, a mechanical stress that the cells do not care about, but it just changes dramatically the nuclear localization of transcription factors like YAP, and that potentially will have an impact on the transcriptome, on the transcriptomic profile of the cancer cells, and probably on their behavior. So the next question was to try to understand whether if these CAFs could actually be dependent on the presence of cancer cells to compress or not. And we also wanted to develop a tool that will allow us to specifically measure the forces that map the forces that these CAFs are exerting. So for this, together with the lab of Stephanie Descroix, and on a very nice collaboration with Marco, that at the time was a PhD student in her lab, we developed these microfabricated structures which are made of polyacrylamide, so they're deformable and they're elastic, and they have beads inside, so we can track them on a just simple spinning disc confocal. And they have a particularity, which is that the pillar itself is not adhesive, so the cells are forced to go to the outside, to surround this pillar, just as if they were around the cluster of cancer cells. And actually, what CAFs do is that they still generate this actomyosin ring, which is just right outside, around the pillar. So what we actually observed, it was that if we compare the same pillar, which in this case is green without any cells, or the pillar when we put the CAFs on it, what we see is that actually there's deformation that happens in this case over time, which indicates that the CAFs are actually able to compress the pillar. This deformation was mapped very thoroughly on a collaboration again with Chavi's lab, in this case with Manu and Ernest, that did just an amazing work that I still have hard time see how we'll be able to do this without them, just by mapping all the single forces that we can actually observe within this pillar, basically showing that the compression happens on the neck of the pillar, and that goes together with some other rearrangement like twisting and shearing of the pillar, and also that goes together with rearrangement of the flat surface that it's around the pillar. So pillar compression is depending again on myosin, as if we put blebistatin, we do not see any deformation anymore. So one of the things, and this takes me to my last part of the talk, is that to compress these pillars, CAFs, they cannot do it on a single cell level, because if you imagine the diameter of one of these pillars, it's much higher than the diameter that one single cell can expand. So they need to coordinate with other cells to do that. And also, we have another experiment that tells us the same, which is if you take this typical CAF cancer cells organization as we have before, and we perform a laser ablation over here, as you can see here, and after we analyze what's happening with the cancer cells underneath, what we see is that 20 minutes after, cancer cells, they have already making these protrusions to try to get some space, and that happens at the area right below where we are cutting. But if we analyze what happens as far away as possible from the area where we're cutting, it happens exactly the same. So that means that the CAFs, they form as a supracontractile unit that are all coordinated together, and the tension that it's actually being released here can be immediately felt on the opposite part. So how CAFs can actually coordinate to do this supracellular activity? First obvious molecule, it was to test what happens with encadherin, which is the main cell-cell adhesion molecule that this guy expressed. But what we actually observed, it was that, at least in the pillar experiment, the CAFs, they can, with or without encadherin, they can exert the same amount of compression. So we next turn into the extracellular metrics, and particularly on fibronectin, which is one of the molecules that we already found that was the most expressed by CAFs compared with normal fibroblasts in colorectal cancer tumors from patients actually. So also another thing that told us to go and search on fibronectin was that if you search what happens with, we see what happens with the mouse model that I just told you before. The controls, well, the formation of this capsule, they produce a high amount of fibronectin, which is highly reduced in the case of the knockout, especially at the areas here where you see the screen cells that are not, they don't express myosin anymore. So contractility, that was known already. Contractility is needed for the fibrillogenesis of fibronectin. So we thought and this together with the local, with the architecture of fibronectin in these CAFs, which make this very long bundles or cables of fibronectin that span several cells. In this case, they span 3 CAFs. So all this data together led us to think that maybe fibronectin could add a scaffold that could add as a mechanical coordinator in between the different calves and allow them to build up the necessary force in the process of compression. So the experiment was to knock down fibronectin, and what we see is that when the cells are not able to produce fibronectin anymore, they're not able to compress cancer cells anymore, and the forces, they go really down compared with control. And this is very important because it's a process that it doesn't happen at a single cell level. So calves, when they do not have fibronectin at a single cell level, they exert the same amount of forces. So it looks that fibronectin, it's important to coordinate cells on a multicellular way to create this scaffold that allows building up force. So of course, we went again on pillars. So these cells are not able to squeeze pillars anymore when they do not have fibronectin, and YAP expression, nuclear localization, gets increased compared with the control. Still not so high as the case of the myosin knockout of blebistatin, because what we think here is that these guys are able to confine, but not to compress the cancer cells. So this takes me to the working model, or to my final slide. I actually have no idea how long it took me because of this stuff. But I hope I was able to convince you that the calves are able to generate this capsule, that it's not able only to confine tumor cells, but also to actively compress them. And this is achieved through this mechanical supracellular coordination that is regulated through actomyosin and also through this scaffold of supracellular scaffold of fibronectin. And that leads to this fork reinforcement, to the transcription factor translocation that we see, for example, with YAP, and that actually is what regulates this capsule formation and the reshaping that we observe in the tumor. So it will be the responsible of the typical microarchitecture that we observe in tumors. So with this, I'm done, and I would just like to thank all the people from the lab, but especially Carlos and Sophie, who have, I mean, of course, Daniela, but who has been working very closely with me, also like people in Barcelona, people in Curie, and also like Pierre and Mathieu, the theoreticians that are doing also like a theoretical model that I didn't have time to talk to you today, and also doctors at Curie that give us samples to work with. And thank you very much to all of you for your attention, and I will be very happy to take some questions.
Thank you, Jorge.
I hope the connection was okay, because I was having a lot this message that your connection is unstable too.
No, we've got you all good. Thanks. We've already got loads of questions, so we'll try and get through as many as we can in the next few minutes. So we'll start with Claire Waterman, who says, are the fibroblasts making and tightening an ECM belt around the tumor cell cluster, i.e. is the contractile belt driving centripetal compression force as opposed to the cast pushing on the tumor cells? And the second question is, and related, is the compression dependent on the fibroblast secreting ECM?
Well, so regarding to the first question, if I correctly understood, what drives the closure of this ring and the centripetal pushing against the cancer cells is just the simple generation of this spur string that will jump on top of the cancer cells. And as it's closing, by friction with the cancer cells, will start generating this little mountain that we see there. And this is the structure that we actually see that will be compressing. And the second question, if the calves, if I correctly understood, it was if the calves, they depend on extracellular matrix to compress. So probably the question was before, I already talked about fibronectin. So yes, yep. They need to generate this this extracellular matrix to coordinate with each other. I don't know if I answered your question, Claire.
Yes. Yeah. The first part of the question was, are the cells pushing or is the belt tightening, right? Like
the belt is tight. The belt is
it's a belt.
Exactly.
Belt
it's a purse string.
Okay, great. Thank you. Minna, do you want to pop in and ask your question yourself? Otherwise, I can read it.
Sure. That was a great talk, Jorge. Thanks.
Thanks.
I'm curious because CAFs have been shown to promote local cancer cell invasion into the stroma, partly through cadherin interactions, which I think is another question in the chat. So how do you think the CAFs might be performing this function if they're forming the supracellular purse string around the tumor?
Well, we cannot really tell you exactly what will happen, for example, with tumor progression or metastasis. We are currently doing that in the lab because the model that we use here doesn't really allow to check for invasion, for example. But what we actually think is that it will be a dual role that will initially kind of constrain tumor growth, and actually it looks like that for what the data that we have so far. But also what we actually think is that if this compression is excessive, it could be the touching little clusters of cancer cells away, as you can actually see in vitro, and generating something that in colorectal cancer is called tumor cell budding. So that will be kind of a secondary effect of what we actually see, and will be rather promoting invasion in this type of tumor. Probably still encadering independent. So I cannot really assess whether the cadherin interaction here is rather important. I can speculate if you want, but
thank you.
Thanks. Hamid Mohammadi asks if the CAF proliferation rate is higher than that of the cancer cells.
It is in this system, just because we kind of putting the CAFs on a kind of a fake situation where they think they're kind of a, I mean, when you just basically put it in culture, they proliferate a lot. So let's say that in vitro, they do proliferate much more than the cancer cells. They do not do so much in vivo. So that's one of the limitations of this study.
Thanks. So I think actually we're going to make this the last question so that we can move on to Minna's talk. But then Jorge, if you want to answer in the chat, or folks can stick around and we can talk more afterwards because there's a few more questions. So from Ricard Alert, is the pressure on the tumor large enough to cause cell death?
Well, I mean, we actually see that there is some, especially in vivo, that there's some apoptosis on the tumor cells. But whether that is directly related to the compression itself, maybe yes, we don't know yet. What we actually know is that if you do not, I mean, if you on a knockout situation when there's no compression, it looks that there's much more proliferation from the cancer cells, which is kind of, I mean, it has been shown in many other situations, right? So confinement will have kind of inhibit tumor growth.
Okay, thanks so much for that. And continue answering questions in another way. So Minna, if you want to go ahead and start sharing your screen. So we're delighted to have Professor Minna Roh-Johnson here as our second speaker today. She got interested in the role of the cytoskeleton in cell migration during her PhD at the University of North Carolina at Chapel Hill. Minna turned her attention to cancer and did her postdoctoral work at the Albert Einstein College of Medicine in New York, and also the Fred Hutchinson Cancer Center research in Seattle. So she's started at the University of Utah in January 2018 as an assistant professor, and her lab uses high-resolution in vivo imaging to study cancer invasion and the tumor microenvironment. So we've definitely got a theme going on today. Thank you so much, Minna, for being here, and take it away.
Thank you. Thank you to you, Jennifer and Adam, for organizing this wonderful seminar series. It's such a good idea to put on a series for the global cell migration community like this, and I'm really thrilled to be part of it. So as Jennifer mentioned, I started my lab at the University of Utah in 2018. So I'm excited to be sharing this story about how we developed the Zebrafish model to understand how cells interact with the extracellular matrix during cell migration. And we've gotten some really unexpected results recently, so we're especially looking forward to getting feedback and ideas from this community. So cell migration is a really critical part of normal development, shown here in these upper examples, but also co-opted in disease states. And questions that really fascinate us in the lab are illustrated by these movies where there are many cells, right, all sending out different signals. There's matrix components. So how do cells still know where to go properly at the right time and to the right place? And who are they interacting with? What signals are they receiving? And because we're interested in those kinds of questions, we study this process in vivo models. So Zebrafish is a great model to study cell migration in vivo. They're genetically tractable. They have large root sizes at larval stages. They're optically transparent, which is great for live cell imaging. Our lab also takes advantage of this highly migratory primary cancer lines, the zebrafish melanoma cell line. It's derived from the zebrafish melanoma itself through a process called Mini Cooper, and it was a technique that was pioneered by Richard White's lab at Sloan Kettering. So these primary highly migratory cancer lines can be extracted and established from primary tumors, and then they can be transplanted into the zebrafish larvae, which allows for same species transplantation, so we could visualize cellular behavior in vivo. So our lab has used this approach to generate our own ZML cells expressing fluorescently tagged constructs to visualize subcellular dynamics of migrating ZML cells. And today I'll be telling you about the work of a really wonderful graduate student in the lab, Chenju, who's been working really hard on a project that's been exciting for us because we think we've developed a great in vivo system to visualize and manipulate focal adhesions, which are the structures that are required for cell matrix attachments. So Chen had began this project by transplanting ZML cells into the hindbrain of 2-day-old zebrafish larvae. This is a good place for injection. The hindbrain ventricle takes up cells readily. The cells will disseminate from the transplantation site all over the animal. And importantly for us, the transplanted cells also integrate into the overlying skin. So these are melanoma cells, and so the skin is their normal microenvironment, but the skin is also planar and close to the surface, which allows us to visualize these cells with high resolution. So if you take these fish then and flip them on their back, an image at the site of transplantation, taking Z stacks from the outside of the animal going in, here you see we're at the outside of the fish, there are those cancer cells that are embedded into the skin layer, and then the rest of the transplanted cells. And in that tissue environment of the skin, cells migrate very readily. Here you can see a cancer cell right at the plane of the skin migrating through that tissue environment. So at first we wanted to use this model to understand how cells migrate in vivo. And we were really interested in learning anything interesting about different forms of single cell migration in different tissue environments. So we generated ZML lines expressing fluorescently labeled ENAVAS. But ENAVAS is the anti-capper that inhibits the function of the capping protein. So it interacts with the growing ends of actin filaments. And because of its role, it associates with areas of actin dynamics. So here ENAVASP is stained in green, and you can see localization at the leading edge in this example. So we transplanted ZML cells expressing ENAVASP into the zebrafish larvae. Here now I'm showing a movie just right at the plane of the skin. We're looking at the ventral surface of a ZML cell. So that's the belly of the cell where it makes contact with the skin. And we were really surprised to see these punctate structures. Some of them are more stable, some of them are turning over. And the reason why we were really excited about seeing these dynamics is, recall that I said that ENAVASP localizes to the leading edge, but it's also been shown to localize to focal adhesions, those sites of cell matrix attachments. And so we wondered whether those structures were actually focal adhesions. So many of you know what focal adhesions are. They've been studied for over 40 years, predominantly in vitro cultures. And because of the great work done by many, we know a lot about the molecular components that comprise focal adhesions, a lot about their dynamics, their force transduction properties. And while they have been predominantly studied in vitro models, it makes it so it's kind of unclear what their composition and regulation and functions are in vivo. And I think a real limitation to understanding focal adhesions in vivo is because they're pretty hard to visualize in vivo and in 3D matrices also. The structures themselves are pretty small, they're transient. So it's not surprising that there hasn't been a ton of work on them in animals. But despite these challenges, there has been a small handful of really beautiful studies exploring focal adhesion dynamics and function in vivo models, and they generally fall into the following categories. The first is sort of live cell imaging of collective migration during tissue morphogenesis. So shown here is an example of apical constriction of amnioserosal cells during dorsal closure in Drosophila, where integrin and talin components of the focal adhesions are labeled in green and red respectively as this process, as the surface of these cells constricts during this morphogenetic process. Another example is during the rotation of the egg chamber in Drosophila, and integrins are labeled here in white. Other approaches are to knock down core components of the focal adhesions and then ask if there are defects in vivo cell migration. That's illustrated by this example by knocking down or knocking out a core component of a focal adhesion, vinculin. And in this example, looking at cardiomyocyte migration during zebrafish heart morphogenesis. So in wild type, the cardiomyocytes migrate up and around the developing heart, and then that migration is inhibited when vinculin is knocked down. And we were able to find one example of single cell migration looking at focal adhesion formation. This example from Anna Huttenlocher's lab looking at zebrafish macrophage migration where Paxillin is visualized in magenta here, another core component of focal adhesions. So I think what these experiments really show is that focal adhesions are really important and likely function during cell migration. But what we were unclear about is whether what were the dynamics and regulations of these structures, particularly for single cell migration in animals, and whether we could use our model to shed light on this process. So when we go back to this example then of these dots that were on the belly of a cell where we knew that area of the skin had ECM, we wondered whether these structures were actually focal adhesions, and if they were, whether we could take advantage of this model to shed some light on focal adhesion behavior in vivo. So first we want to determine whether these were focal adhesions. So the first thing we did was ask whether the cells that we transplant in at the skin are actually in contact with extracellular matrix. We did that by now I'm flipping the view of the zebrafish. This is now a lateral view of zebrafish. We're looking side on and using immunostaining techniques against laminin, shown here in magenta, as well as live cell imaging with collagen, shown here in magenta, and the cancer cells in green. We saw with light microscopy that cancer cells are making contact with extracellular matrix. We also did sagittal sections of those animals and looked with transmission electron microscopy at the ultrastructure. And what I've outlined here in these TEM images is the white dotted lines is the matrix of the skin. And here, this slightly pigmented cell is a zebrafish melanoma cell, and you could see that it's making contacts with the matrix of the skin. And furthermore, if you zoom into that region of contact, you can see electron dense structures where the cell is making closer contact with the matrix versus its adjacent areas that resemble adhesions in other systems. So from these results, we think that yes, these zebrafish melanoma cells are making direct contact with matrix. So if they are, can they then transduce force on that matrix, right? So a key component of focal adhesion biology is that it attaches to matrix and then it pulls on it so it can propel its cell body forward. So to address this question, we got the collagen line from Paul Martin's lab and then made transgenic animals from it where the collagen is labeled with GFP in green, and then the Z-mel cells are shown here in magenta. What you'll see in this movie is this Z-mel cell will migrate downward, and what you'll see with the collagen fibers is the collagen fiber sort of getting pulled toward, like buckling toward that cancer cell as it migrates. So here's the merge, where you can see the cancer cell is migrating down. And as it's migrating down, these collagen fibers, maybe it's easier to see in the green only channel, are buckling toward it as the cancer cell slides past it. So it suggested to us that cells, the cancer cells can transduce force to the outside matrix when they're migrating along them. And we're following up these results now with intracellular tension sensors to quantify intracellular force transduction. Okay, so because we originally used ENAVASP to visualize these punctate structures that we hypothesized were focal adhesions, we wanted to follow up on these studies with a core bona fide focal adhesion marker. And we chose Paxillin, which are represented in these orange circles in this schematic. So Chen made Z-mel lines with that Mini Cooper technique expressing Paxillin GFP. And what we saw, this is actually a movie that I showed you earlier. I've just slowed it down now. And now we're just looking at the ventral surface of that cell. So that cell that's making contact with the ECM. What I hope you can see is that when cells are extending out protrusions in the direction of migration, that you see these Paxillin positive structures, you can see them better here, that form underneath the leading edge as that cell is actively migrating. So again, these results in combination with the ECM results suggested that these structures were focal adhesions. And then we really wanted to use our model to understand how these focal adhesions were forming in vivo and whether focal adhesions are fundamentally different in vivo than what has been described in cell culture. So we first performed FRAP analysis to dissect Paxillin binding kinetics. Here first are the Z-mel cells plated in 2D. You can see in this little red circle, we photo bleached the region inside the red circle and then look at the recovery over time. We performed the same experiments with Z-mel cells transplanted into the animal, where we photo bleach the region and look at its recovery over time. And here's the quantification. And what we saw were that Paxillin positive focal adhesions exhibit a faster recovery rate in vivo than they do in vitro, as indicated by the T1 half. We also looked at lifetime, which is the amount of time where we get full initiation of focal adhesion assembly and then full disassembly. The Paxillin lifetimes actually didn't differ between the cells in vitro versus in vivo, but the assembly and disassembly rates did. So we can take the slopes of these lines to look at focal adhesion assembly rate and disassembly rate. And what we saw is in vivo, the focal adhesion assembly rate was faster, and then it's, for disassembly, it slowed down. So what this suggested to us is that Paxillin regulation, focal adhesion formation and turnover might be differentially regulated in vivo than in culture. So to test this hypothesis, we decided to look at the regulation of Paxillin. So Paxillin is a scaffold, it's phosphorylated on a number of residues, tyrosines and serines, that's shown here as these little red circles. But there are these two residues that have been shown to be key sites of regulation for cell migration and focal adhesion turnover. And so we want to determine whether these sites were differentially regulated in animals versus in cell culture. So we use a phospho-specific antibody against the phospho version of this tyrosine 118. And we saw that Z-mel cells in culture nicely localized phospho 118 Paxillin at sites of where focal adhesions are, as expected, because this had been shown in mammalian systems that this site is phosphorylated, and it's important for the disassembly of the focal adhesions, and therefore it plays a role in cell migration. However, when we looked at these same cells in culture, in zebrafish, we saw something very different. So these are immunostained now zebrafish, where the Z-mel cells have been transplanted, that's in green. And if you look at the phospho 118 staining, we couldn't see it in the Z-mel cells. We think that our immunostaining worked just fine because tissue resident cells stain positively, and we now know that these are neutrophils. We also imaged the developing heart of the zebrafish because that's also positive for phospho 118. So it was really interesting to us to see that this key site of regulation of Paxillin is absent in vivo and suggested that Paxillin might be regulated differently in vivo than in vitro. So to functionally test whether this residue was important for cell migration, we exogenously expressed variants of that site, either mutated to a glutamic acid to serve as a phosphomimetic, or to phenylalanine to serve as non-phosphorylatable version, and then exogenously express them in these zebrafish melanoma cell lines. Again, these experiments have been done in mammalian cells, and what has been shown in mammalian cells is that expression of this non-phosphorylatable form inhibits cell migration, because again, you can't turn over the focal adhesions. When we express them in Z-mel cells in culture, we see that each of the variants localize to focal adhesion structures. And so we went ahead and then transplanted them in to zebrafish and asked, how does that affect cell migration in vivo? And what we saw here are representative movies of the wild type Paxillin, the phosphomimetic, and the non-phosphorylatable. And I hope what you can see is that the non-phosphorylatable version of Paxillin, those cells exhibited faster migration than the phosphomimetic and wild type. We're still, Chen just got these mutants a couple of weeks ago, and so we're still acquiring and quantifying these movies. Here's what we have so far in terms of cell rates, where we visualize faster migration in the non-phosphorylatable version of Pax versus the phosphomimetic and the wild type. And again, this was really surprising to us because previous literature had shown the non-phosphorylatable version of Paxillin in vitro exhibits slower migration. Okay, so today I told you a story about how we developed a model that gives us a really good look at focal adhesion biology during single cell migration in animals. We showed data that suggests that these structures are focal adhesions because the Z-mel cells are physically interacting with matrix, they localize core components of focal adhesion machinery, and we have evidence that they also transduce force to the surrounding matrix. Furthermore, with this model, we think we've uncovered some novel aspects of focal adhesion regulation in vivo. We show that focal adhesion assembly and disassembly rates are different in vivo compared to in vitro, and that a key site of Paxillin regulation, this residue 118, as identified in cell culture studies as inhibiting cell migration, the non-phosphorylatable inhibiting cell migration, in vivo, we see that this version has the exact opposite phenotype. And we're really interested in following up, looking mechanistically to explain these observed differences, as well as developing approaches to spatially manipulate focal adhesion function to test their role in single cell migration. Okay, so with that, I want to thank the lab, especially Chen, who did everything that you saw today. She's been really wonderful, working really hard on this project. Our community, the wonderful Zebrafish community, is always very generous with strains, our cores, and funding. And I'm happy to take any questions.
Awesome, thank you so much. As usual, folks, drop questions in the chat, or you can ask them yourself. So our first one is from Oscar Ruiz, who says, as you know, the zebrafish epithelium is made of 2 layers. Are the injected Z-MELs staying in between the periderm and basal layer of the epithelium, or are they below the basal layer?
Yeah, it's a good question, which is why we did the TEM, because we couldn't tell with the light microscopy. So it appears that the majority of the transplanted cells that integrate into the skin are just basal, underneath the basal layer of that double layer. However, in zebrafish, that's also where the melanocytes are, and the melanocytes are the cell that gives rise to the melanoma. So at least in the zebrafish system, that's where we would expect the native environment to be, although we understand that that is distinct from a mammalian skin where it would be in between the epidermis and dermis.
Thanks. Next question is from Carlos Perez Gonzalez. He says, great talk. To what extent is the difference between in vitro and in vivo Paxillin turnover dependent on substrate stiffness? From the ECM displacements in vivo, ECM seems soft.
Yeah. So we assume that the ECM in the zebrafish is softer than glass. And we also wondered whether that could play a role in terms of the absence of phosphopaxillin staining, because it had been previously observed that stiffer substrates have reduced phosphopaxillin staining. We can't say that that doesn't play a role. It probably does, but we don't think that's the whole picture. And the reason is because other cells that are in the exact same environment, so neutrophils and macrophages, appear to follow the same paradigms. Now, we're still following up on those studies by driving these Paxillin mutants specifically in neutrophils and macrophages. But it suggests that substrate stiffness isn't the whole story. The other reason why we think it's not the whole story is when you change the stiffness, that usually changes the lifetime of the focal adhesions. And that we didn't observe either. We saw changes in the rates of assembly and disassembly, but lifetime stayed the same. So we think that while stiffness may be playing a role, it's likely that whatever other molecules are then localizing to those structures in vivo may be playing a role also.
Thanks. From Rito, we have the question, I'm not really familiar with the Z-MEL system. Could you explain what kind of cell numbers you get in a specific area? I ask because in the images, very obviously the cells are much bigger, and I'm wondering if their apparent scarcity might have a role in the way they behave.
So like maybe I could break that down to a couple questions. So the Z-MEL system is the adult zebrafish forms a melanoma. And that was a transgenic animal that formed a melanoma by driving a constitutively active BRAF mutation in a p53 minus background. After the adult forms the melanoma, the tumor is extracted and then sorted for just the cell population, and then the primary line is made, and that's the Z-MEL cell. I think, and please tell me if I didn't misinterpreted the question, that you're saying that the cell density may affect the cell migration. So a lower cell density injected might change how the cell migrates. This is something we've also looked at. As far as we can tell, there's no difference in cell migration or cell rates according to the number of cells that are transplanted in. It is true that the representative image that I showed you with the non-phosphorylatable version looked like a higher cell density, but what we actually noticed in that line is they also proliferate faster, which we've been unable to really explain yet. So there are a couple different phenotypes with that, with those mutants.
Thanks. The next question, we're going to skip the next one because that was already answered, but Brendan Whitelaw says, very cool. I may have missed this, but do more heavy-handed ways of disrupting focal adhesions affect migration in vivo?
Yeah, so we have done pharmacological inhibition where we add the RGD peptide or we add a FAC inhibitor. Those prevent the cells from even attaching onto the skin in the first place, which makes sense because we inhibited the focal adhesions. And so they do impair then subsequent cell migration and dissemination over the body, but we think it's because we inhibited their ability to even integrate into the tissue. We are following up with the spatial inhibition of focal adhesions so they could first attach, and then we could inhibit their functions to ask how they affect subsequent cell migration.
Great. From Anna Pasapera, so in mammalian cells, in 2D and 3D systems, tyrosine phosphorylation of Paxillin correlates with focal adhesion assembly, and phosphorylation of focal adhesion kinase at tyrosine 397. So did you look at the focal adhesion kinase tyrosine 397 phosphorylation?
Yeah, it's a great point, and we haven't. And the only reason why we haven't is antibodies are notoriously difficult to get working in zebrafish. It's sort of the thing that zebrafish people don't talk about. A lot of times because those antibodies were made for mammalian systems and they don't cross-react as well, but also some challenges with the zebrafish system. So we have been unable to get a lot of the antibodies to work for these phosphospecific antibodies like the FAC397, as well as the Paxillin, the other Paxillin residue that I didn't talk about today. I said that there were two and we looked at one because that was the one that we could get working. So one of the ways that we're going to address this, because if we can't get the antibodies to work, is to make the mutants of those versions, right? So you can make a phosphomimetic of the FAC397 and the non-phosphorylatable version. Chen has also already made the double mutant in the Paxillin for those two tyrosine residues. And so that is how we're going to tackle whether those proteins and then those residues have a role in vivo compared to in vitro.
Just quick question from me. Are the so the Paxillin residues that you mentioned in zebrafish, are they the same ones that are important in mammalian cells?
Yeah, so it's actually the tyrosine residue 118 in mammalian cells. It's when you look at the sequence in zebrafish, it's the same residue, but it's actually residue, I think like 95. It's just a different place, but if you look at the surrounding sequence, we are able to identify it.
Okay, it's that 118 that's special.
Yep, exactly. Yeah.
Cool. And a question from Chitnissa. Do ZML migrate? Do ZML cells, I guess, migrate as single cells or also tend to migrate collectively?
Yeah, so we've been looking pretty hard for collective cell migration because we think this could be a great system to study collective cell migration. As far as we can see in the skin, they mostly migrate as single cells. However, when they locally invade into tissue, they do begin to divide and they stay attached to their sister cells. So we're hoping that we can use those examples as examples of collective cell migration through tissue. It may not be then at the skin with focal adhesions because cells that are surrounded by tissue, like neuroepithelial tissue, for instance, take on a very different form of migration. But I think that will be really interesting to look at.
Great, thanks. And then I think this will be our last question, unless anyone else wants to pop in, from Emrinder Nain. Great talk. Is there any evidence that the size of the extracellular matrix fibers can alter focal adhesion size and assembly?
Yeah, so trying to think if we have any evidence for that. You know, we have not been able to manipulate the ECM pore size or the thickness of the collagen bundles. So I can't say that anything about, you know, manipulating and then having some observed effect on focal adhesion biology, except to say that I think it very likely will affect how a cell migrates. You know, if you're too dense and you need to migrate through, you probably won't use adhesion-based migration and might switch to a different form like a non-adhesive based migration. But we have not been able to experimentally test that in our system.
Okay, thanks very much. I think that's the end of those questions. So thank you very much, Minna. That was really great.
Great, thank you.
And thank you, Jorge, as well. Jorge, did you want to answer? I don't know if you got a chance to answer the remaining questions in the chat, or would you like to answer them now?
Not in the chat yet.
Okay. Should we go to? I don't know how many we had left over.
So.
We've got
I mean, yeah, I have them up here. I think if we want to go back, and I'm not sure if the people who asked the questions are still here, but there's just a handful of them, so we might as well go through. So I think the next one was from Oscar Ruiz. Did we ask this? Have you tried any mosaic analysis with the calves? What percentage of calves would need to have functional actomyosin or fibronectin to exert force?
Not really. I mean, in this case, we use kind of a very, I would say, homogeneous population of calves that will all have the ability of producing ECM, and they're highly contractile. But that's a great suggestion. I think just by trying to understand which is the percentage of fibroblasts, because I don't think that we'll need that all the fibroblasts they will need to be so highly contractile. So probably with a lower percentage, and I think just getting this number will be pretty interesting. Yeah, that's a nice question.
Thanks. I think we missed Anthony's question. Maybe we've covered it already. Work from Javi's lab a few years ago showed that ECAD, NCAD heterotypic interactions promote epithelial cell migration. Have you looked about whether that exists in your system? And if not, is it due to compaction? Do these tumors still express ECAD or any ECAD area?
So yes, the tumors, I mean, calves, they express NCAD, and the primary, I mean, the cancer cells, they do express ECAD. It's true that when we knock out myosin in the calves, and YAP gets into the nuclei, we see a reduction on the ECAD and the cancer cells, and also there's some cells that can undergo EMT. But NCAD, it looks that it's still important in the system. I mean, I didn't want to enter into the details with this because I didn't have much time. But so when Xavi's lab showed that when you have one calf that goes against a cluster of cancer cells, it can establish heterotypical contacts with the cancer cells and pull them out of the tumor. And so in this case, the system is so crowded with fibroblasts that they cannot really exert this single cell pulling force. But instead, what they do is that once they are generating this contractile ring that will be the one that will be compressing the cancer cells and slides on top of the cancer cells. For this compression to happen, cells need to friction against each other, and we actually know that Encadering is one of the molecules that is responsible for this friction from the calves with the cancer cell. So if you don't have encadering, you're still able to compress the pillar because the pillar is kind of a huge structure. But if you do not have encadering on a 2D flat surface, the cells do not friction anymore with each other, and the compression doesn't happen anymore. So basically, the ring just closes on top of the cancer cells as if it was non-adhesive substrate. So still encadering, it has a relevance here, but I didn't want to mention this.
Okay, thanks for that. Just real quick, just to tell you, Minna, a comment from YouTube, great talk, and hi from Vancouver from Esther Verhagen.
Awesome.
So,
hi, Esther. Thanks for tuning in. She was my undergrad research advisor.
Oh, nice. Okay. Well, yeah. So we have folks over on YouTube who can't really directly interact with you guys. So nice to be able to pass on messages from them. Okay. So I think we have 2 more questions, if you don't mind, Jorge.
Yeah, sure.
Just since you had your technical troubles, it's nice that we can go back.
I'm sorry.
No, I mean, hey, it's the world we live in. So from Samaya, do the squeezed cancer cells come out, and do they form a circulating tumor cells that come across the blood stroma? So I think you've touched on this a bit already, but
yeah, that's a great question. I hope I would be able to. I mean, I wish I was able to answer it. So no, I mean, that's something that I'm actually wanted, I mean, now in my lab, I wanted to try to understand what are the functional relevance of this compression. What we actually think is that the release of circulating tumor cells could also be controlled by the active compression of CAFs, but so far I have zero data related to that. So, but that's a great question.
Thanks. And last question for today is from Si Chang, who says, have you compared the behaviors of CAF and normal fibroblasts in a cancer-free system to see whether normal fibroblasts can also squeeze?
So, we've tested like CAFs and normal fibroblasts from colorectal cancer patients. So those are human fibroblasts, and we tested it in the pillar system, and it looks that normal fibroblasts, they squeeze much less the pillar because they're less contractile, obviously, but they still do so. So, but on a much reduced level.
Okay.
Go ahead,
Thanks,
Jen.
guys.
No, thanks, you guys.
Yeah, I was just gonna sort of sign off and thank you so much both to Jorge and Minna for those wonderful talks, and thanks for kind of squeezing everything into the shortened time period. We really appreciate that. So we'll see you guys next week. We just have a handful more scheduled for this season. So